I'd like to welcome you to this year's installment of the Richard A. Scott Lecture presented by the Center for Genetic Medicine. The Richard Scott Lectureship was established by a generous gift to promote or to recognize the accomplishments and the contributions of researchers who have contributed to genetics and medicine. And this year's speaker is Tom Cech. Now, Dr. Cech needs very little introduction, but I'm kind of required to give one, so I'll keep it short. Dr. Cech is extremely well known in the field of RNA biology and in many other fields as well. He received his BA in chemistry at Grinnell College in 1970, and then his PhD in chemistry from the University of California, Berkeley in 1975. Since then, he's accumulated 16 additional doctorate degrees, honorary degrees from other institutions, and that's just the beginning of his accomplishments. He did a short postdoc, only 2 years, in the lab of Mary Lou Perdue at MIT from 1975 to 1977, and then he joined the faculty at the University of Colorado-Boulder, where he has been ever since. He did terrific research for which he was recognized, and he was made a member of or an investigator of the Howard Hughes Medical Institute, and he continued his association with the Howard Hughes Medical Institute, becoming president in 2000, and he was president through 2009, and he has now returned to being an investigator at the Howard Hughes Medical Institute. And so it's appropriate that today we're having this talk in the Hughes Auditorium. The recognitions that Dr. Cech has received are just amazing. The Nobel Prize in Chemistry in 1989 are really just a drop in the bucket. I have never seen a CV that has had so many awards that they have to be grouped by decade, and the entire list takes an entire page. So clearly, Dr. Cech has done terrific work, making contributions that have been recognized by at least 40 or so different prestigious institutions. I won't begin to describe them. I'll just say it is our honor and pleasure to have Dr. Cech giving the Richard Scott Lecture today. Thank you.
Well, thank you, Greg, for that introduction. I recognize most of what you had to say. I think it was me, and I'll check up with you about a couple of the points later on. It's always great for me to be back in Chicago. I was born here. My parents moved to Iowa City, Iowa when I was 5 years old, which I think was a good move, but then for the whole rest of my childhood, every Easter and every Thanksgiving, we would come back to Chicago to spend it with the relatives, and we would always go to the Museum of Science and Industry. So I grew up being stimulated by that fabulous museum and also the Field Museum, which is not too far away from there, occasionally the Shedd Aquarium. And those were really formative experiences in my growing up. Another formative experience in Chicago was the semester I spent at Argonne National Lab, which drove me out of physical chemistry. But sometimes, for those of you who are still in the early parts of your career, you know, having an experience that tells you what you don't want to do in the future is just as important as having an experience that tells you what you do want to do. And then, what else about Chicago? I first met Hannah Gray here when she was president of the University of Chicago, and that turned out to be influential because then she called me up later and asked me if I wanted to be president of the Howard Hughes Medical Institute, which was an exciting decade in my life. But I've given that up now, and so this is the transition I've made just recently from this wooded, cloistered, sort of CIA safe house-like environment of the headquarters of the Howard Hughes Medical Institute in the Washington, D.C. area to this place where this is my view from my laboratory window at Colorado. And if you think sun and snow don't mix very naturally, maybe in Chicago that's a little less common, but it snowed. It was on Friday, it was 80 degrees in Boulder. Over the weekend, it snowed, and now it's 70 again. So this actually is very common to have this sort of transition, in the spring especially. At least at the University of Colorado in Boulder, when we think about the central dogma of molecular biology, you know, the DNA makes RNA, makes protein, we have a little different view of the relative importance of these molecules than maybe some of you have been taught. And so we really love this big green one in the middle. And part of the reason for this is that RNA shares with DNA the ability to carry information. And you all know this. In fact, there are viruses like the common cold virus, like the flu virus, that don't even mess with DNA. Their genome is RNA, and they just go RNA to protein all day long. So clearly RNA can be a good carrier of information. But what we have learned more recently is that RNA also shares with protein the ability to speed up biological reactions. RNA can be quite a good biocatalyst. And that work on the catalytic RNAs was really the major thing that was going on in my laboratory during the 1980s and 1990s. But just as life is sometimes thought to have evolved from an RNA world through a ribonucleoprotein world to the present world where catalysis is dominated by protein enzymes, our laboratory has sort of made this same evolution as well. And so we no longer work on these pure RNA catalysts, but instead we're trying to understand the structure and function and the roles of the RNA in ribonucleoprotein catalysts. Because it's really biologically a much richer system. These RNP enzymes, many of them are central to our own biology. The ribosome, of course, being the most glorious and fantastic example responsible for message-encoded protein synthesis in all of extant life. But we don't study the ribosome. Instead, we're studying this enzyme telomerase, which is responsible for extending and helping replicate the ends of linear chromosomes in eukaryotic organisms, including humans. And the telomerase was discovered by Liz Blackburn and Carol Greider in 1985. And they also found that the telomerase had an essential RNA component. And they went on to show that one of the functions of that RNA was to template the sequence of DNA nucleotides that were added to the end of the chromosome. So there's a stretch of this RNA that has this template function. You can see it's about to finish off this synthesis of this telomeric repeat when it adds another T and another G. And then the primer, the DNA, slips back and repairs in this region, this alignment region, and then gets extended again. And so the enzyme can make these hexanucleotide repeats over and over again from a single binding event. So it has sort of a processive extension of the DNA end. And the sequence that's shown here, this T2G4 repeats, that hexanucleotide, is from the Tetrahymena thermophila, ciliated protozoan telomerase. That was the first telomerase that was discovered. Interestingly, that same ciliated protozoan was the one in which we found the first example of a ribozyme, a catalytic RNA. So this has been a very important organism for sort of discovery biology. But the human sequence is really very similar. Again, it's a 6 nucleotide repeat, and it's repeat of the sequence TTAGGG instead of TTG, GGG. So similar sort of concept, just a single nucleotide change in the template RNA is all you need to change that telomeric DNA sequence. So because I thought that this is a medical center, is that right? Are some of you actually see patients? Raise your hands if you're involved in, Larry, Larry, you still see patients? Oh, okay, well, so I thought that it might be interesting to you to hear about some of the disease associations with telomerase. And the short story is that the concentration of telomerase in our cells has to be maintained at a very specific amount, because there are problems that have been demonstrated from either having too much or too little. So it's very tightly regulated. The older story, which I'm not going to talk too much about, is that in most human somatic cells, telomerase is turned off before we're born and remains transcriptionally, the gene remains transcriptionally inactive. Now, this is the gene for the protein component, not the RNA component, but there's also this protein component, which is a reverse transcriptase. And I'm sorry, I'm usually better organized than this. Let me just go back to say that this RNA cannot do all this job by itself. It requires an active site made of protein, and this is the telomerase reverse transcriptase. And this TERT was discovered by Joachim Lingner when he was a postdoctoral fellow in my laboratory in 1997. So it's this reactivation of this telomerase reverse transcriptase in human cells that is a step towards formation of diverse human cancers. And it's not just a surrogate marker or something that comes along for the ride. It's actually a causative step in oncogenesis. And that's been shown both in the positive direction. You can immortalize normal cells by transfection of TERT, and it's been shown in the negative direction that you can quickly kill human cancer cells by putting in, for example, a dominant negative version of the TERT, of the gene for the TERT protein. But the more recent story, which is more complex, is that having too little telomerase has been associated with a number of diseases of tissues that have to continue to proliferate even in the adult. So tissues such as skin, fingernails, gut epithelium, the lining of the lung, these tissues require constant renewal, and having even haploinsufficiency. So if you're a plus-minus heterozygote for the telomerase reverse transcriptase, that's enough to have problems in maintaining the stem cells that are required for continued proliferation of these tissues. So the clearest example is this rather rare genetic disease, dyskeratosis congenita, which has been really firmly nailed as being a disease of telomerase insufficiency. There are mutations, which I'll show in a minute, in the TERT protein component, in the RNA component, in discarin, which is an accessory subunit that helps in the assembly of the telomerase, all of which have been firmly associated with the disease and seem to account for pretty much all cases of dyskeratosis congenita. Now, the rest of these examples are different. These are disease associations. I'm not saying that all aplastic anemia or all idiopathic pulmonary fibrosis is due to a telomerase problem, but there's been some fraction of these diseases, some probably minority fraction, that has been associated through family pedigrees. And it remains to be seen, and one would in fact expect, because this is such a fundamental housekeeping protein, such a fundamental housekeeping RNP unit, that one would think that this would be even more widely important. So just to give so So I separated here and said that, you know, cancer is having inappropriate overexpression of TERT. Telomerase insufficiency causes other problems, but you see leukemia shows up on the bottom group as well. And even more interesting, in the dyskeratosis congenita patients, there is this incredible propensity to have early onset cancer, such that about half of these individuals get cancer by age 50. And you can see that it's quite a range of solid tumors. And you say, well, why, if telomerase is sort of pro-proliferation, would there be a problem with cancer if you have too little telomerase? Well, it has these dual functions, and we'll talk about this some more in a minute. It's not only important for chromosome replication and cell proliferation, but also for genome integrity. So because telomerase makes the sequence that caps off the ends of our chromosomes, having too little of it can cause a lack of protection of the end of the chromosome. And then you can get genome instability, chromosomal rearrangements, which can be pro-oncogenic. And so it can really go in both directions. So I promised to show you some mutations that had been seen in dyskeratosis congenita families. And you can see that throughout the RNA subunit of the telomerase. Now, just to orient you here, this little part of this human telomerase RNA is the template that is determining the sequence of the telomeric DNA. All of the rest of this RNA has other functions, such as recruiting the TERT into the complex, recruiting accessory proteins into the complex. And mutations in multiple positions that destabilize this RNA structure have been associated with disease. Similarly, in the reverse transcriptase protein subunit, throughout the entire length of the gene, there are mutations that have been associated either with aplastic anemia, dyskeratosis congenita, the pulmonary fibrosis, AML, etc. And just to orient you to, I'll be coming back to this TERT protein, so I'll just say a bit about the domain structure. This part, called the RT domain, has very definite homology to other reverse transcriptases, such as HIV reverse transcriptase. So this makes sense. What does telomerase do? It uses an RNA template to direct the synthesis of DNA. What does HIV reverse transcriptase do? It uses an RNA genome to make a DNA copy, which can be integrated into our chromosomes. And so it makes sense that biochemically, this would be a reverse transcriptase, so it's not surprising perhaps that it has the same hallmark sequences that form the active site for reverse transcription. But in addition, there's a portion of the gene here that is not found in HIV reverse transcriptase, that is an RNA binding domain, because unlike retroviral RTs, this protein carries around its own RNA all the time, so it has a domain that's responsible for RNA protein association. And then it has an N-terminal domain shown here, which anchors the DNA primer and keeps it engaged with the active site of the reverse transcriptase. And again, this is specific to telomerase reverse transcriptases, not found in other RTs. And as you can see, mutations in all of these domains and also in the C-terminal domain have been disease associated. Now, we don't do too much work in this area, but we did have an opportunity recently to collaborate with Steve Artandi, who is in the medical school at Stanford. And and this paper is about to come out in Nature in about the next month, so I'll give you an advanced look at it here. So he was generating the induced pluripotent stem cells from patients that had this dyskeratosis congenita, to see the question being, to what extent can we recapitulate the disease phenotype on a single cell level by starting with cells from fibroblasts from these patients and now turning them into pluripotent stem cells. And what he found was that you could take cells from patients that had different forms of the dyskeratosis congenita, an X-linked form, an autosomal dominant form, which is the haploinsufficiency that is due to mutations in the telomerase reverse transcriptase gene. This is a slower onset form, a gradual failure to properly extend the telomeres leads to a very slow inability to maintain the proliferative state of the cells, and that he could recapitulate these the phenotypes from the patients at the single cell level, the extent to which the telomeres shortened, the extent to which the cells lost their proliferative capacity, and then found that the quantity of active telomerase in these cells was in fact exactly correlated with the severity of the disease. And so what was our role in this? Well, of course, in order to make this argument, you have to know the level of telomerase activity that is caused by these mutations. And so we did the enzyme assays on these telomerase mutant enzymes after expression in human embryonic 293T cells. So we expressed the mutant TERT and also the RNA, then immunopurify the complex that's been assembled in the cells, and look at the effect of a single amino acid change on the activity. And here you can see, maybe just look at the right-hand part of this. Here's the wild-type telomerase. I'm actually not going to describe these 4 different conditions under which the activity was measured. Here's a single amino acid substitution. A non-synonymous SNP in the TERT gene causes a 90% reduction in telomerase activity. Certainly not dead, but it's just a small fraction of the wild-type activity. And the same is true of this other single amino acid substitution. And so this is the team that we've assembled to sort of start looking at these disease variants of the TERT gene. We're using mostly undergraduates. So this is Christina, Caitlin, and Jesse are the 3 undergraduates. We recruit them pretty young at Colorado, but these, in fact, have not been recruited yet. These were children of some of the other hikers. And so what Jesse, one of the undergraduates, has been doing is looking through the dbSNP and other databases at what single nucleotide polymorphisms are found in the TERT gene. Some of these have been associated with disease. And then these days it's so easy, you can take an undergraduate from an intro chemistry lab into your research laboratory. Within a week, they've made site-specific mutations. They have confirmed by complete sequencing the fact that there's only 1 mutation in the gene. They've expressed this, reassembled telomerase, and done an enzyme assay, which 20 years ago, this would have been a whole PhD thesis for somebody to have been able to do this. What you see in these ladders is we introduce a short telomeric oligonucleotide. So this is just an oligo with the sequence TTA, GGG, TTA, GGG. And then radioactive guanosine, deoxyguanosine triphosphate, alpha P32 labeled, so that when it's incorporated, the products are radio labeled. And then again, you can see here at 2 different primer concentrations the activity of the wild-type telomerase. Here's a polymorphism that is fairly common in the human population that some people have said they thought was disease associated, but others have said it was just a polymorphism which had no phenotype. And at least in our assays, under these particular conditions, it looks like it has full activity. But other single amino acid changes caused by single nucleotide changes have quite a severe reduction in activity. And in some cases, such as these two mutants, it's pretty easy to understand structurally why these amino acids cannot be substituted without a deleterious effect, because here's the active site in the reverse transcriptase domain. Here's one of these mutations was a mutant of lysine 902. Here's another one of arginine 865. And you can see how both of these amino acids are nestled right next to the active site aspartic acids, which are holding on to the catalytic magnesium ion, which is important for catalyzing nucleotide addition. So in these cases, it's pretty clear that these are directly involved in active site and supporting the formation of the active site that does the chemistry of telomerase. And so it's not surprising that these mutations would be deleterious. So now I'd like to switch gears and move from the telomerase to the telomere itself, so to the chromosome end, which is the substrate, the natural substrate for the telomerase enzyme, but is also essential for genome stability. In fact, I'll give you an example of that shortly. So the very ends of our chromosomes, as shown in this in situ hybridization by Peter Lansdorp, there's something common to all of the ends of all the human chromosomes. And what this is, of course, is this repeated sequence that I've already mentioned, this TTA-GGG. But what I didn't point out is that much of the end of the chromosome is double helical DNA. Just like you probably are saying, well, isn't all DNA double helical? Well, much of it is double helical. The TTA-GGG is paired with CCC-TAA repeats. But at the very end, the last 100, 200, 300 nucleotides of our chromosomes are a single-stranded region. And we give a very technical term to this. We call it the tail at the end of the chromosome, okay? So this single-stranded overhang or tail is important for a number of reasons. First of all, it recruits some proteins that are single-strand DNA binding proteins that help stabilize the end of the chromosome. And secondly, it provides the substrate for the primer for telomerase extension. So that the telomerase has to be able to base pair DNA to this RNA template, and if the RNA template were already, if the DNA primer were already part of a double helix, it would be, if it would have its own antisense present, it wouldn't be able to pair to the telomerase. So I'm going to talk a bit about the properties of this protection of telomeres, protein number 1, which was discovered by a postdoctoral fellow, Peter Baumann, in the lab about 9 or 10 years ago. And this is very common now to the way that many genes are being found in humans. And that's by using databases and bioinformatics and relying on data from model organisms to sort of work your way to the human gene. So people had been wondering for a long time whether there was a human protein that would bind to the very ends of human chromosomes. And there was a precedent for this because ciliated protozoa, again, going back to Tetrahymena and some of its relatives, definitely had a telomere end binding protein, but no human counterpart had ever been found. So Peter was looking through sequence databases, and he found a fission yeast protein sequence, a Schizosaccharomyces pombe sequence that looked like it had a little bit of similarity to the ciliated protozoan genes for the end-binding proteins. And so in POMB, it's very easy to do a genetic test. So even if the idea doesn't seem to have a lot of legs to stand on, you can test it and refute it or support it very quickly. So he made a heterozygous diploid for this gene that we now call Pot1, and then sporulated the cells, and the haploids, 2 of them grew up into nice large colonies. Those were the Pot1 plus haploids, and 2 of them formed these really micro colonies, and these were the Pot1 minus. Well, this, of course, doesn't show that this has anything to do with telomeres. It just shows that this is a gene that's important for cell growth. But before I go on, let me just say that this phenotype is very different from what happens when you knock out the TERT gene in Schizosaccharomyces pombe. So here, you find that the TERT plus and the TERT1 minus colonies are indistinguishable following germination of the spores. In fact, they have enough of a reservoir of telomeric sequence that it takes about 75 population doublings before the failure to replicate the chromosome end accumulates enough that the cells have critically short telomeres and stop growing. So telomerase has this interesting property that it's essential in the long run and completely dispensable in the short run. Whatever this Pot1 protein is doing, it's clearly important early on. You don't have to wait very long before you see an effect on cell growth. Well, the real excitement came when Peter looked at the state of the telomeres in these Pot1 minus cells, and they didn't have any telomeres anymore. They were completely gone. So here's the homozygote, here's the heterozygote, Pot1 plus over Pot1 minus. Still, this distribution of sizes reflects the fact that the telomere length is not maintained at an exact size within cells, but over a range of sizes. Then the 4 haploid spores germinate into cells that give either no telomeres if they're Pot1 minus or regular telomeres if they're Pot1 plus. What turned out had happened was that most of the cells can't protect their telomeres at all. They're all very quickly degraded, and then the chromosome ends are unprotected, and they start, I won't show you the evidence for this, it's all published, but they start fusing to each other, giving dicentric chromosomes, and all of those cells die because they have a fused chromosome with 2 centromeres. And then the cells that are lucky enough to fuse the left end of chromosome 1 to the right end of chromosome 1 to form a circle, and the same with chromosome 2 and chromosome 3, are the survivors, and they grow up without having any telomeres and still being able to go through vegetative growth quite well. They have a lot of trouble going through meiosis. And then the biochemical properties of this protein were also consistent with a role in binding to the very end of the chromosome, because the POMB protein bound only to the single-stranded telomeric sequence, the G-rich strand, not to the complementary C-rich strand, so it had sequence specificity. And if you hybridize these 2 together to make double-stranded telomeric DNA, the protein completely ignored it. So it was specific for single-stranded DNA of telomeric sequence. Peter quickly was able to use sequence databases to jump to the human ortholog of this POMB protein, and the human Pot1 protein also had this property of binding specifically to the G-rich strand now of the human telomeric sequence and not to the double-stranded DNA. A number of people then started exploring the biology of Pot1, and just to make a long story short, there were 2 different types of observations. A number of laboratories found that it was involved in telomerase regulation, and a number found that it was involved in protecting the ends of the chromosomes and preventing them from being seen by DNA damage machinery as a place that should be joined or repaired, but instead to be ignored by the DNA damage machinery, which is a good thing for the natural ends of chromosomes. So when you delete or use siRNA to knock down the Pot1, all of a sudden the chromosome ends are perceived as sites of DNA damage that need to be repaired. Not something that you really want to have happening at the natural ends of your chromosomes. Then Ming Li came to our lab, trained at Steve Harrison's lab at Harvard as an X-ray crystallographer, and very quickly was able to form crystals of the POMB protein in a complex with its single-stranded telomeric DNA partner, several different crystal forms, some of which diffracted to atomic resolution. And here in this electron density map with the model of the DNA and the protein fit into the electron density, you can see that there's this wonderful stacking arrangement where bases stack on a base from the DNA, stacks on an aromatic amino acid from the protein. And this turns out, I point this out because it turns out to be sort of a defining feature of this class of proteins, this base-base amino acid stacking. Here's a ribbon diagram showing what's called the oligonucleotide oligosaccharide binding fold, this highly twisted beta sheet, which is provides the surface for DNA binding of this for the telomeric DNA of the Pot1 protein. And here in this electrostatic representation, you can see that this is a highly electropositive surface that's interacting with the negatively charged DNA, single-stranded DNA. Now, I almost said RNA. That's partly because I have RNA on the brain. But for those of you who are used to looking at nucleic acid protein complexes, this looks weird for DNA protein, right? DNA is always this very regular double helix. This actually looks more like an RNA protein complex because once you quit constraining a nucleic acid to be part of a double helix, it can take on a wonderful myriad of shapes and forms. And in fact, the nucleic acid is all wadded up and interacting with itself in this cleft of the protein, as well as interacting with amino acids of the protein. So then Ming went on and solved the structure of the human Pot1 single-stranded DNA complex. And here we see two oligonucleotide oligosaccharide binding folds. So this is not a dimer of the protein. This is two domains of the human Pot1 protein, one of which recognizes one repeat of the human telomeric sequence, TTA, GGG, the other of which recognizes the next repeat. So each protein unit, in this case, recognizes two telomeric repeats. And again, as before, this motif of having the base-base amino acid stacking, base-base tyrosine, base-base phenylalanine. Here's an interesting case where there's a stack of four. Three of them are amino acid side chains, aromatic amino acid side chains, and one is a nucleic acid base. And so much of the stability of this interaction comes from this base stacking array, but the specificity comes not so much from that, perhaps not at all from that, but rather from amino acids on the edge of the beta sheet poking into the nucleic acid and making hydrogen bond donor and acceptor interactions that are specific for each of the bases. So here you can see that if you tried to change this thymine for cytosine, for example, that you would have the donor and acceptor pairs for the interaction with that threonine all messed up. So the actual recognition comes from this hydrogen bonding network. So the final picture then is that this Pot1 protein buries the DNA in the single-stranded DNA in this DNA binding cleft. If you have a long single-stranded region, you have a bunch of these Pot1 proteins, they can coat the whole single-stranded DNA. I'm not going to show you that picture, but it's very nice. Every 12 nucleotides binds a protein unit. And this seems to be really a good solution for chromosome end protection, right? If I wanted my chromosomes ends protected, this is exactly what I'd want to do, is to bury that end so that it can't be seen by nucleases or by the machinery that is engaged in DNA damage, DNA repair, non-homologous end joining, recombination. It makes it invisible. However, now we've got a bit of a conundrum because telomerase has to be able to find this end. This is the same DNA that has to base pair with the RNA template of telomerase. So how can you have, you can't have your cake and eat it too. So what's good for chromosome end protection is perhaps really bad for telomerase accessibility. So this is what we've been working on now especially in the last 3 years. And we still don't have a solution to this problem. The first thing we thought is, well, let's just test and see if our idea from the structure is correct. Does this structure really turn off telomerase completely? Or maybe we're missing something. And so when we formed the Pot1 DNA complex, and I won't go through all of this data, but just to say that if we put the Pot1 down here near the 3' end of the DNA, sure enough, just as predicted, it almost completely inhibits, 99% inhibits telomerase activity. On the other hand, if we put the protein upstream from the end so that there's still a 3' extension that can pair with the telomerase RNA template, now it's quite a good substrate. So having the protein close by is not bad. Having the protein at the very end, in fact, does hide the DNA from telomerase as we would have predicted. Well, so then we were going to continue studying this question of, well, then what is it that slides the protein over? But then 3 other laboratories at this point found that Pot1 had a heterodimeric partner protein called TPP1. So we had been looking at only half of the story. There's another protein that sandwiches between the Pot1, and if this is the Pot1 and this is the DNA, there's another protein that sandwiches from the other direction, makes the interaction even tighter, so we might expect that things would get even worse, but instead when we use that pair of proteins as the binding partner to the telomeric primer, now we get this hyperextension. Telomerase really likes that as a substrate. And it took us quite a bit of time and a graduate student's entire thesis to come up with the following model for what in fact is happening here. So first of all, Chris Alatrick, who's now got her PhD, found that the Pot1 TPP1 complex, first of all, helps recruit the DNA that's bound to telomerase. Then after the binding event, it decreases the falling off of the DNA primer from telomerase so that the synthesis, which normally has fairly low processivity and only goes a few rounds before dissociation, now is much more processive. And we've not proven that it makes this little inchworm extension, but that's our current model, is that the Pot1 TPP1 is interacting with the telomerase, and that as the DNA is being extended, this interaction continues to inhibit the dissociation of the primer and thereby encourage further elongation. And that's why we get this very long ladder of extension products. Well, so we showed this biochemically, but at least in our lab, we always like to show things both biochemically and genetically. So the question is, well, if this is true, maybe there should be mutants of the TPP1 protein and of telomerase that inhibit this interaction and thereby prevent this hyperextension from occurring. And so we've made some progress in both of those directions. So first of all, we were able to get a crystal structure of one of the domains of the TERT, of the telomerase reverse transcriptase. We marched along the surface of this protein and made single amino acid changes, and we found one that had exactly the expected effect. So I don't know if I can take you through this or whether you should just take my word for it. In any case, in each group of 4 lanes, the 4th lane is adding the Pot1 TPP1. So here for wild type telomerase, you again can see that the extension becomes hyper elongated when you have both of these telomeric binding proteins present. However, this one mutant, G100 changed to a valine, has no extra extension when you add Pot1 TPP1 relative to what you get with Pot1 bound to the end of the chromosome by itself. All other mutants, even ones that decrease activity, still show a response to these telomere proteins. So here's a single amino acid mutation. It's down in activity, but when you add the Pot1 TPP1, you still get this increased processivity of the extension. So we think we may have located a surface on telomerase, which is involved in this putative interaction. And then how about from the point of view of the TPP1 protein? Here we've again marched along the surface, mutated single amino acids, conserved amino acids, from whatever they are naturally to usually an alanine. Again, the idea here is that it's the surface of a protein that is most likely to be engaged in intermolecular interactions. And mutations there are the least likely to sort of disrupt the core function of the protein. So what we were looking for here were mutations that did not prevent the protein from engaging in this chromosome end capping complex. We wanted that to be wild type, but we wanted a mutation that would affect this next order of interaction with telomerase. And so all of these surface amino acid mutations pass the test of not affecting the affinity for the Pot1 DNA complex. So here's a binding curve of just the Pot1 protein binding to single-stranded telomeric DNA. When you add TPP1, you increase the affinity of this interaction about 7-fold, and that's true of all of the wild-type TPP1 and also all of the mutant ones. But you can see that their response to the telomerase enzyme assay falls into sort of 2 categories. A number of them, like wild type, still give this very processive extension when you add Pot1 TPP1 mutant to the DNA and now extended with telomerase. But others show still core extension, but not this increased processivity. And so here's the quantitation. Some of them are right around the positive control, so this part of the surface of the protein is presumably not involved in this interaction. Here are a few of them, like these two, this one, that are almost as bad as having no TPP1 around at all, and these are good candidates then for amino acids that are making an interaction surface. So I know all of that is hard to follow, and I'm not sure if this is going to make it harder or easier, but this is this is a dream or a speculation, okay? So what we've done here is to take the little bits of crystal structures that different labs have determined for different parts of this big complex from different organisms. And we've sort of, to the best that we can, Dave McKay in the laboratory has stitched all of this together. He's very nervous about me even showing this. But I think it's very useful for at least, I mean, immediately when he tried to build this model, he said, but where do I put this? Or how do I know this? So it's very useful in terms of phrasing all of the big list of questions that we don't understand yet, right? So what we have here is the active site of the reverse transcriptase. We take this from a crystal structure published by Skordalakis from the red flower beetle, because that's the only one that we have to work with. So that's this aqua sort of structure. You can see the RNA template region here, and it's base paired to the telomeric DNA. Well, the Skordalakis structure didn't have the telomerase essential N-terminal domain, but we determined that structure from tetrahymena, and we know it has to be linked to the rest of the reverse, so that's just shown here as a dotted line. And then we have that mutation, the G100V, that affects the ability to interact with the telomere protein, TPP1. So look at this. It's pretty close to this acidic patch on TPP1 where our other mutations are that affect the process. Well, how do we know where to put the Pot1 and TPP1? Well, we have crystal structures determined in our lab, determined in Ming Li's lab, of the Pot1 bound to DNA, of the TPP1, and the relative orientation of the two. Since we don't have a co-crystal structure that has all of this in it, we use the ciliated protozoan crystal structure from Steve Schultz to orient these two relative to each other. So now you can see here's the chromosome end binding to the chromosomal DNA, but that at the same time we propose is interacting with telomerase through the interaction between these two regions. The chances that this is right in detail is roughly 0, but the chances that it's sort of the spirit of it is right, I hope there's a good chance that that could be true. And so in close, so that last work was done by J.K. Nandakumar, postdoc in the lab, working with undergraduate Caitlin Bell, who's been in the lab ever since she was a freshman. And I would like to thank former laboratory members who contributed to the telomere and telomerase project through the years, a number of which now have their own faculty position. Joachim's back in Switzerland, but also faculty positions here in Chicago, in Kansas City, University of Michigan, or Johns Hopkins, or Texas A&M, and then the current group, including Dave McKay, who helped with the modeling, and Art Zog, who continues to help with just about everything in the laboratory. And finally, since I have a captive audience, I'm going to spend just 1 minute in unashamedly advertising a new graduate program that we're starting. Now, most of you are probably not looking to go to graduate school as I look out at this audience. But I'll bet many of you mentor or have in your labs undergraduates, right? Or maybe you have children who are ready to go to graduate school. And so we've started this bold new graduate program to complement our interdisciplinary research activities at the University of Colorado-Boulder. And I call it IQ Biology, that stands for Interdisciplinary Quantitative Biology. Instead of bioscience with attitude, it's bioscience with altitude, since we're at 5,600 feet. And the students come in without having an arranged marriage to any discipline or department. So they come in with an interest in applying computation or engineering or physics or chemistry to problems of biology. They take 9 months of a course which is topics in interdisciplinary biology, taught by myself and a physicist. And then they do laboratory rotations in at least 2 different disciplines. And then after 9 months, they choose a discipline, and then they take an abbreviated, slightly abbreviated set of disciplinary requirements, and they get a PhD in the discipline and a certificate in IQ biology. So it's a new way of thinking about training of interdisciplinary graduate students, and we've just enrolled our first class. We've got very good students. I don't know how we deserve this because it's a brand new program, but we got some very good students to come for the first year, and they'll be starting in August. So if you know any students, they have almost a whole 8 months to log on to this website and apply for to be part of the 2012 class. And I thank you for your attention.
So Dr. Cech will answer questions, but before we have the question and answer session, there's a few people that we have to thank in addition to Dr. Cech and Richard Scott, who provided for this presentation. We need to thank Dr. Peter Kopp, who is the interim director of the Center for Genetic Medicine, and some of the staff of the Center for Genetic Medicine, Nell Wasserstrom and Megan Boyle, and the rest of the people in the center who are helping out with microphones and various aspects of this. So let's give them a round of applause. Dr. Cech will now take questions.
So one specific question, one more general question. One specific question is, we talked this morning about those other RNA binding proteins, since your lab is also working on FUS.
The long non-coding.
Right. Does that family interact with any components of telomerase, especially whether FUS binds to, with any affinity with TAGGG? Repeats.
I'm sorry, do any of what other?
Of the FUST DP43 family.
No.
You have tested that, and that's not binding.
No, well, there's just there's no evidence for there's no genetic or other evidence for it. We have not directly, we didn't have a reason for that hypothesis, and so we did not test that. But we could, it would be a good control for our experiment.
I'll give you some reason later. And then a more general question, perhaps not so much related to this particular lecture, is could a ribozyme or any of those telomerase, for example, component could exist in 2 different energy status at the same time, which is more a physics question than biochemical question.
I'm sorry, could a ribozyme exist in 2 different entities?
Energy status.
Energy
status at the same time. Maybe there's no answer to that question,
Well,
or what's your view on that?
Yeah, well, within the population, there
I'm talking about single molecule.
single molecule.
Any evidence for or against?
I don't know. Again, we could talk privately, but I don't know of any such evidence.
Thanks.
Certainly, in a population, this happens with RNA.
A single
happens
molecule.
all the time.
That's what I'm. Thank you.
Yes. I think they want to run up and hand you back a microphone so that.
Yes, I read a paper in Science, I think it's 15 years ago, on what wonders were going to come out of telomerase, both in killing cancer cells and in lengthening life. And it was a definitive article. And here we are 15 years later, and I've seen nothing.
Yes, I mean, even more pointed, there was a whole book written called Fountain of Hype. That was about the discovery of telomerase and the perceived medical insufficiencies, the fact that we haven't really come through on this. So with respect to the cancer end of things, there's certainly been good proof of principle that if we could inhibit telomerase in cancer cells, this would be a wonderful anti-cancer approach. However, getting an actual compound that does this efficiently, the industry has spent tens of millions of dollars just trying to get a lead compound good enough to put into clinical trials. And there's one compound in clinical trials, a small phase 1, phase 2. I think the problem is this is such a slow enzyme. It doesn't have to add very many nucleotides per cell cycle. Even if you inhibit it for a whole cell cycle, it can catch up the next cell cycle. So I think that may make it a bad target for chemotherapeutic compound. Anyway, it has the short answer for the cancer story is that nothing good to report on that end. The other end of the coin, the telomerase insufficiency causing early aging, I think the best way to look at that is it's not really premature aging of the whole organism. So if we could rejuvenate our telomerase, I don't think it would make us live longer, because I think a lot of our mortality is due to accumulated oxidative damage of both our genome and all of our proteins in our cells. So I'm not sure having more telomerase would help that, but certainly there are these clear cases now of premature aging of certain tissues, bone marrow particularly, but also skin and nails and gut epithelium and lung, et cetera, where clearly being able to ramp up the telomerase in those tissues would be therapeutic. I think there's good reason I would believe that. I mean, it's so clearly correlated. 50% reduction in activity is enough to give the disease, so if twofold increase would be enough to cure it. But that's not so easy to do, right? A twofold upregulation of a complex RNP enzyme like this. We can do this in yeast. We can cure this problem in yeast, but to do this in a mammal would be difficult. So I'm not holding my breath. I think these are going to continue to be disease associated, and we're not going to be able to really do much about it.
Tom, is there any reason or evolutionary reason for the generation of the telomere sequence the way it did, like T, T, G, 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 G? Why not any other sequences? Or is there any sequence preferences that actually evolved that allowed the sequence to be a telomeric sequence?
Well, there are many papers written ascribing special structural significance to these blocks of G-rich sequence. My lab has actually worked on that, and these structures do form. They're called G quadruplexes. That could be important. On the other hand, the sort of minimalist view would be that once you have a particular telomeric sequence and now evolve proteins that are critical for genome stability that recognize specifically only that sequence, that you're sort of frozen in to a particular combination of telomere sequence and binding proteins. And once you're in that happy situation, it's hard to now move over and say, oh, just change the telomeric sequence. That's easy to do. We do that in vivo and in vitro, and it's deadly. But for the trivial reason that the capping proteins can't bind anymore, and so you get genome instability. So I think once you find a happy situation of a successful telomeric sequence that binds these end-protecting proteins, you're stuck in that situation, and it's hard to move to a new sequence. That's the frozen accident theory.
Well, let's thank Dr. Cech again and move to the reception.